脑人言。
也非常荣耀。周健老师其实是我们那个老人研这个 community 的这个一起就是 members 在一起的老师，也是就是我们也是老人研，就是每个时间段，然后我们都会邀请到非常精彩的报告，然后我们也参与这个主持和这个汇报自己的这个科研成果之中。然后今天非常有幸，周健老师亲自来，就是不仅是作为这个主持者，也作为一个就是来那个来交流自己科研的这样一个非常好的一个形式。然后希望我们这场报告通过交流这样一个深入交流，能够一个对周健老师的这个工作的更深入了解，一个是刚刚希望他说的这个 MR 实验室建立的实验室，也希望大家如果有兴趣的小伙伴愿意去他那里做博后啊，或者做 PhD 啊，都可以和他联系。然后如果你感兴趣他这个研究方向，还有这个自闭症这个神经遗传学的领域的一些专家，还有一些老师同学，如果大家感兴趣，可以在会上进行交流，或者我们会下还可以进行和周健老师进一步交流。那我先简单介绍一下周健老师的这个简历吧。周健老师现在是这个 Emory 就是艾默里大学这个医学院人类遗传学和儿科双聘助理教授。那他本科毕业于武汉大学生科院，也是我的校友，因为我本科也是从这里毕业的。2017年呢，他在德克萨斯 Texas A&M 获得博士学位，师从生物钟及昼夜节律领先驱 Paul Harding 教授，随后去了贝勒医学院从事博士后研究，师从世界顶级神经遗传学家，大家都应该是耳熟能详的那个霍达让比教授，MECP2 的克隆和发现者，然后专注于这个神经发育疾病的分子遗传基础，并在大脑中首次发现全新的这个染色体复合组，也是今天周健老师要介绍这个工作，MECP2 TCF20 complex，阐述了这个它在神经发育和致病中这个自闭症谱系障碍中的重要作用。周健老师在博士期间呢，获得非常多的荣誉包括这个登凯 NRR 的那个朗比学者奖，还有院士杰出学者奖，NIH 的 K99 和 R01 的奖项。我们知道其实这个 K99 R01 是非常，就是美国读过博士后小伙伴都应该知道，这个非常难拿的一个就是 funding，它也是通往这个就是 independent 的这个 PI 的这样一个非常强大的知识，所以它是一个 NIH Pathway to Independent Award。然后周健老师以第一作者的身份呢，发表了多篇文章及评论，包括这 Science、PNS、Gene and Development、Current Biology and Plus Genetics，并获得了很多基金的支持。那么周健老师的研究方向呢，主要是集中在这个自闭症谱系障碍，然后英文就是这个 Autism Spectrum Disorders，简称 ASD。我们知道它是影响儿童社交交往和学习沟通的这个很严重的神经发育疾病。那美国的疾控中心的最新筛查数据显示，每36名儿童之中就有一个患自闭症，所以这个疾病它是一个非常有待于就是科学家们集中攻关，并且去克服一个很严重的一个疾病，能够改善这个儿童的这个生活质量，并且减轻社会的这个经济负担。所以自闭症的这个我们知道自闭症其实它是一个遗传性疾病，但是它和其他遗传性不一样它就很多这个就是 mutation。组合，它这个成因就非常错综复杂，非常就是至今也没有一个非常明确的这个理论或者一个学说能够去把它解决或者阐述清楚。所以说这个 science，在几年前公布的这个125个科学问题里面，它是一个非常重要的一个问题，也是一个最前沿的有待科学家去攻关的一个问题。然后雷特综合症呢，就是英文是 Rett syndrome，它是一个非常典型的自闭症谱系障碍之一。主要是由 MECP2 基因的突变而生，这也是后来让米教授他们这个杰出的这个发现。然后这个 MECP2 呢，它会这个突变呢，它导致这个疾病的具体的分子机制呢，也目前呢就尚未可知，就是不是很清晰。所以周健老师呢，在这个他的工作这个大脑中首次发现了全新的这个染色体复合物 MECP2 TCF20 这个 complex， 阐述了这样一个复合物。它怎么样在其中这一系列的自闭症谱系障碍中的这个重要作用，并且建议出了新的这个自闭症基因和发病机制。那么基于这些研究，周健老师的课题主要现在就是进一步采用这个小鼠和神经干细胞， 相关模型呢，致力于研究神经发育疾病的遗传分子及其神经机理，并开发新型的非侵入性的治疗手段。那么今天就是周坚老师将汇报的这个工作的题目呢中文是自闭症谱系障碍的分子遗传基础，然后他将用英文向我们就是汇报这样一个相关的工作呢，英文题目是呢 The Molecular Genetics of Autism from Bedside to Bench and Bank。从基础到临床，我觉得这个是非常有意义的讲座，大家可以看一下这个就是这个基础到临床转化是怎么样一个思路过程，觉得这个是有在这个讲座中就是我们可以学到的东西，所以下面我就把这个舞台交给周倩老师，来给大家交流一下这些相关的这些工作啊，谢谢。
好的，谢谢陈老师的介绍，然后同时也感谢脑人研的邀请，给这个机会让我跟大家一起交流脑科学和相关的疾病。那其实我就这一阵，我发现我觉得还是用英文来讲吧。一个原因是因为正好我在准备一个其他的国际会议，就偷个懒一起准备了。另外一个是。大家也知道很多那个名词很难给它翻译成中文，所以说最后变成一个中英夹杂的东西那很多人不太习惯，所以说我就干脆就用英文来讲吧，希望大家可以理解。So let's get it started, all right? So thanks again for inviting me, and it's a great pleasure to be here and share with you our research. So I'm a molecular geneticist, and I'm interested in understand the molecular basis of different type of behaviors are regulated in both health and disease conditions, particularly autism spectrum disorder. So today I will share with you how we are going back and forth between humans and animal model to study the mechanism of the disorder. So I want to start by asking you to imagine the daily life of a student, right? So we woke up in the morning, we go to classes to learn new skills, and then after school, we hang out with our friends and families in the bars, in the restaurant, the karaoke's to enjoy the rest of the day. Now, for children with neurodevelopmental disorder, such as autism spectrum disorders, all this daily behavior become a challenge. So why is that? Well, this is because these behaviors are regulated by the activities of neurons in our brain. Now, at the molecular level, there are many transcription factors that orchestrate expressions of thousands of genes in these neurons, and that give rise to those behaviors. This process often go wrong in diseases such as autism. So really the goal of our lab is to study the mechanism of how these biological processes are regulated in order to understand the disease mechanism, and also in a hope to develop a new treatment that could potentially help the patient. So what I like to do today is to first give you sort of a big pictures of the genetics and molecular basis of autism. First, talk about the genes, then about the mechanism of the disease. Well, in the second half of the talk, I will share with you what effort we took to move towards therapeutic, which could potentially help the patient. So let's start with the problems. So autism spectrum disorder is a disease that affects how children learn, interact with others, and their social activities. And many children with autism also have hyper or hypoactivities, responding to external stimulation and environment, meaning that they might see and feel the world in a different way than many of us here. Now, further complicated situation is that many autism, especially those so-called syndromic autism, they have co-occurring conditions, comorbidities, such as intellectual disability, anxiety-like behavior, and many of them actually have sleep problems. So why it is important to study autism? Because it is quite prevalent. And now we know that autism is diagnosed in 1 in every 36 children in the United States and many other countries. And that is costing us over $100 billion only within the United States to deal with the disease. But even that, there is no targeted treatment because we don't quite understand the mechanism of the disease. Now, I think the major challenge for autism study is perhaps the huge heterogeneity, as we know that over hundreds of genes has been found associated with these conditions. Now, to get to understand all the causes of autism at once mechanistically is very tough. So what we decided to do then is to start with a distinct form of autism called syndromic autism, where many features constantly occurs in patients with the same disease. So this will give us a handle to then connect to other syndromic autism and further expand the network. And this would allow us to find a converged pathway that allow for the development of a new treatment. So for that, we decided to study a disease called Rett syndrome. So what is Rett syndrome? The Rett syndrome is a devastating neurodevelopmental disorder. It is also one of the most frequent syndromic autism found in girls, while they usually have normal development for the first 1 or 2 years of their lives. And then they were followed by a regression, meaning that they will lose the language and skills they learned in the early days, and they stopped interacting with their parents, they stopped responding to others, and besides autistic features, they also have other comorbidities such as intellectual disability, language delays, and hand stereotype, as you see in the figures that the girl is rubbing her hand, and that is a pretty typical Rett symptoms. They also have anxiety-like behavior, ataxia, seizures, tremors, meaning the shaking of hands, and many of them actually have sleep problems. So you can imagine how devastating it is for the patient, and especially for the parents, because they just spend the best 2 years of their lives with the kids, and then they will start to gradually lose all of those. Now, the genetic cause of Rett syndrome was found back in the 1990s by Dr. Huda Zogby, and now we know that more than 95% of the Rett cases are caused by the mutation in a gene called methyl CG binding protein 2, or MACP2 for short. And you can probably tell from its name that this is a DNA binding protein. It binds to methylated cytosine through the MBD domain, and it will recruit its partner, the Encore complex, through another domain. It's called a TRD domain. So not surprisingly, these two functional domains are the hotspot for missense mutation found in the Rett patient. And that has led to the hypothesis that MACP2 is a bridge between DNA and the Encore complex. And for quite a long time, we think this is the only function of this protein. Now, if that is the case, you could imagine mutation disrupts DNA binding, showing in the middle of the figure, or mutation disrupts Encore binding, shown on the right of the figure. They should have equivalent outcome. But when we look at the data from mouse studies or patients, this hypothesis is not really supported. So I'm giving you one example. So this is the surviving curve of a mouse, and you can see the black line on top is pretty high throughout the time because that's the healthy animals and they live quite long and healthy. Now, if you look at the dashed line and many other lines, they are dropping, and they are dropping because the animals are dying, and they are dying because they are carrying different type of MACP2 mutations. And the dashed line showing the mouse carrying the now allele, meaning total loss of the genes, and they die pretty quickly. Now, if we look at the red line, this is representing the mice carrying the MBD mutation, disrupting the DNA binding. And we can see they also die very quickly, comparable to the now allele, suggesting that binding to DNA is very important for MACP2's function. If it cannot bind to DNA, it cannot do anything. Now, the interesting part is, if we look at the blue curve, representing mice carrying the TRD mutation that completely abolished Encore binding, they actually live much longer. And we kind of see the similar result in humans, while patients carrying these TRD mutations, they turns to have a milder phenotype. So what this tells us, it tells us that this NCOR complex is not the only important functional partner, and there might be other important interactors of MACP2 that contribute to the disease. So this really motivated us to then search for those new interactors, and we took a proximity-dependent approach called BioID, where we fuse MACP2 with an enzymatic domain that will label any nearby protein with a biotin. We can then purify those proteins, do mass spec, and find what they are. Now, because binding to DNA is very important for MACP2's functions, here we also used two negative controls that either block its access to the DNA or block its access to the nucleus. So these would allow us to specifically looking for proteins that interact with MACP2 at the chromatin interface. And those proteins are more likely to be the functional partner of MACP2. So using this approach, we identified a handful of candidates, and today I will focus on the top two, the TCF20 and PHF14. So TCF20 is short for transcription factor 20. This is a transcription cofactor. It has transactivation domain, a DNA binding domain, and the PhD domain on the C-terminal for protein-protein interactions. The second candidate is the PHF14, and it gets its name by having multiple PhD finger domains. So both of these two proteins are either DNA or chromatin binding factors, which are exactly what we're looking for using this approach. Now, before we dive into the biology, first we searched the database and we found these two proteins potentially form a complex with two other proteins. But this complex has never been reported in the brain. And to find out whether these exist in the brain, we did a co-immunosuppression experiment. As you can see here, we pulled down each component, TCF20, PHF14, and RAID1. Well, in each cases, you can see the rest of the complex came down together. So this really shows for the first time that this new protein complex exists in the brain. Now, what's more important is that human genetic data showed this new complex is genetically associated with multiple syndromic autism. For example, mutation in TCF20 causes autism and another disease called the TCF20 associated neurodevelopmental disorders, or TEND. Now, mutation in the other gene called RAID1 causes a Smith-Magenis syndrome. This is another childhood neurodevelopmental disorders. Now, both of these two conditions, they share certain features we see in the Rett patient, such as autism, intellectual disability, and sleep disturbance. So what this tells us, it tells us that this complex is perhaps very important for brain functions because mutations in each component would cause some sort of a brain damage. And we're quite excited about this finding because this is exactly what we are looking for at the beginning, a group of disease that are molecularly associated. Now, of course, the first thing we want to do is to validate the protein-protein interactions. And here, I'm showing you a cell-based assay. Well, in the mouse embryonic stem cell, MACP2 will be enriched to this methylated heterochromatin foci, and this is marked by the very bright green spot here. And if we co-express PHF14 protein, we can see they followed MACP2 and also relocate to this foci. And this is MACP2 dependent because without MACP2 protein, you see there is no such effect. So this suggests a strong physical interactions between the two proteins. Now, if we do the same for TCF20, which is the top candidate, we did not see this effect. So what is going on? Well, it turns out that the PHF14 is important for this interaction because if we co-express all three proteins, now we see they all relocate to this foci, suggesting that PHF14 is actually mediating the interactions between MACP2 and the rest of the complex. And we further mapped the interaction domains that are important for protein-protein interactions. And just to make the long story short, we found the PhD finger domains on the PHF14 interact with MACP2, well, the MBD domains of MACP2 is important for its interactions with PHF14. And I will come back to that when I talk about patient mutations. So now we have a new complex in the brain. What is the biological functions? Now, first, we want to know where does this protein expressed? Okay, so I did immunostaining for TCF20, which is the key component of the complex. And you can clearly see that it specifically expressed in neurons marked by these neural end markers in green. Next, we want to know what about other proteins? So we co-stained TCF20 and MACP2, and we found there is a nice co-localizations between the two proteins in the same neurons. So this is consistent with the finding that they actually form a complex in the same cells. Now, what are they doing in these neurons? So from previous study, we learned that too much of MACP2 in neurons will cause synaptogenesis problems. So those neurons will have a lot more synapses marked by the co-staining of the pre and post synaptic markers pointed by this white arrowhead. So we hypothesized that this phenotype is potentially mediated by the interactions between MACP2 and its important partner, the TCF20 complex. And if that's the case, we could reduce the amount of this partner, and that could potentially rescue the phenotype. So this is what we are trying to test. So we cultured mouse neurons in wild type and with overexpressions of MACP2. And indeed, we found too much of MACP2 increased synaptic density, which is quantified on the right, showing in the red bar. Now, if we use shRNA to knock down the expressions of TCF20, this phenotype gets fully rescued. Now you see the green bar is comparable to the wild type controls. Okay, so this suggests a strong genetic interactions between MACP2 and TCF20 complex in regulating the neural morphologies. Now, this data motivated us to then test it in vivo using animal models for behavior. And to do that, we generated a new mouse line, the TCF20 deficient mouse line, with a loss of one copy of the genes. This is to mimic the patient with missense mutations or loss of function mutation with TCF20. And we found that the mice actually represent many of the features we've seen in the patient. So here, showing on the left, the mice are significantly smaller than the wild-type litter mice, representing the developmental delay of the patient. And we also seen multiple neural behavior deficit, including altered sociability, impaired learning and memory, and anxiety-like behavior. Okay, these are all seen in the patient with TCF20 mutation. Now, to study the functions of this protein in combination with MACP2, we cross this mouse line to the MACP2 overexpression mouse line, and that produces 3 different genotypes. So these 3 genotypes are actually matching the 3 types of neurons I showed you before. So we are actually doing the same experiment, but now in vivo using animal and behavior as a readout. So we did a series of behavior tests, and here I'm just showing you one example. This is called the open field test, while the animal will be put in an open box to freely explore the areas. And if the mice are anxious or they're nervous, just like many of us, we are going to hide in the corner, right? And if they are less anxious, they're more willing to explore the center area. So what we found is that overexpression of MACP2 in these animals reduce the time they spend in center, suggesting that those animals are more anxious, that don't want to explore in the center. Now, if we genetically reduce TCF20 in these animals, again, we see the phenotype gets fully rescued. So we also look at other behavior deficits, including learning and memories, social abilities, and found indeed, many of these features are rescued and partially rescued by the genetic interaction, suggesting that now this new protein complex is important to regulate the mouse behavior and potentially in the patient as well. So the next question is, how does this contribute to disease? Now, for that, let's move from bench to the bedside. And if you recall, several components within this complex are associated with neurodevelopmental disorders. Now, given that PHF14 is an important mediator, we want to know whether mutation in this gene also cause disease. So we reach out to doctors and human geneticists worldwide, and indeed we found a few patients with mutation in PHF14, and they're suffering from autism, intellectual disability, speech delays, sleep problems, which are similar to Rett syndrome. And we found actually very interesting point mutations from one patient that map to the MACP2 interaction domains on PHF14. So we hypothesized that these point mutations might disrupt the interactions between the two proteins. And just to test that, we did the same cellular assays expressing PHF14 with these point mutations. And now we see that PHF14 protein carrying this point mutation is no longer recruited by MACP2 to the foci, suggesting that these point mutations completely abolish the interactions between the two complexes. So we're quite excited about this finding because it seems that now we found out the mechanisms of why these patients carrying these mutations have these neurological features. So we recently made a mouse model that carrying exactly the same mutation, and we're trying to characterize it at multiple levels to further study the functions of this complex in a disease context. All right, so now we have a complex. It's important for neuromorphology, it regulates mouse behavior, and it contributes to human diseases. Now, of course, the important question is, what's the molecular mechanism? Well, because TCF20 and MACP2, both of them are transcription regulators, we want to look at the downstream gene expressions in the animal models. So we look at RNA-seq data from each of these two mouse models, and we found indeed hundreds to thousands of genes are misregulated. And we found there is a strong and significant overlap between the two. So for those genes that are shared between these two regulators, there's a high concordance, meaning majority of them change in the same direction with similar amplitude in both animal models, suggesting that they are likely co-regulated by these two proteins. So we next want to know, what are those genes? What do they do? So we then did the gene ontology analysis together with the network analysis, and we found important neuronal pathways such as synaptic pathways and ion channels are misregulated and enriched in the downstream genes. We also found important developmental pathways such as Wnt signaling pathway and cell migration pathway are enriched, suggesting that this pathway potentially contribute to the disease pathogenesis. And we further look at the mechanism of this regulation, and for the sake of time, I won't show you all the data, but we also found that the whole complex actually binds to the promoter of these genes and regulating their expression. So all this data suggests that this new complex binds to the chromatin and regulate important neuronal and developmental pathway. And this potentially contribute to the disease we see in the patient. Now, to summarize this part, I showed you that we found a new TCF20 complex that physically interact with MACP2 and that mediate MACP2-related autism spectrum disorders. We found disruption of this new protein complex actually underlines a wider group of distinct forms of autism. And at the molecular level, we found this complex co-regulates many important neuronal and developmental pathways that potentially contribute to the disease. All right, so I hope I convinced. So, well, back to our original questions. Well, indeed, using this approach, we are able to find a group of disease that are molecularly associated. Now, in the future, using the same strategies, we can expand this network and further study the complexity of autism. So based on what I showed you, I hope I convinced you that by studying the biology, we can learn the mechanism of the disease, and sometimes we can make new diagnosis. Next, I want to show you another story and show you how we can use what we learned to design new therapy to actually help the patient. So this story started a few years ago. Well, this boy named Henry reached out to us, and because he was diagnosed with Rett syndrome, and he had a typical Rett phenotype, such as lack of communication with his parents. He has developmental delay, hypotonia, means change of a muscle tone, and he also have a motor dysfunctions. And we sequenced his genome and found, not surprisingly, there is a point mutations on his MACP2 genes, the G118E, which is mapped onto the DNA binding domain. And just to confirm that this point mutation is causal to his features, we made the mouse model that have exactly the same mutations, the G118E. And we characterized the mice and found, well, indeed, it represents many of the features we see in the patient, such as social deficits, learning problems, anxiety-like behavior, and motor dysfunctions. And here, the arrowhead is pointing to his hind limb, which is holding all the time, and that is a typical neurological features we've seen in the rat animal models. Now, because this point mutation is mapped onto the DNA binding, we want to know whether it disrupts DNA binding. So we did a chip assays and found, indeed, the mutant proteins significantly reduce the binding of MACP2 on the targeted proteins, targeted genes. We also look at global binding and found a similar result, right? So what's important here is that although this mutated protein have reduced DNA binding, it does not completely abolish its ability. So it still binds to DNA with about 30 to 40% of the capacity. So that really gives us a hope, because if we could find a small molecules or drugs that increase or enhance the binding of these proteins, we might be able to help. And to do that, we need a way to quantify the DNA binding of these molecules in real time. So for that, we collaborated with Robert Tijan's lab at UC Berkeley using their single molecule tracking technology. So how does it work? Well, we can label MACP2 with the halo tag. So this is a very bright fluorescent signal, and that allows us to visualize the movement of a single molecules of MACP2. And you can imagine the DNA or chromatin are very heavy and mass biomolecules. And if a MACP2 binds the DNA, it will stay there. It won't move. And for the diffusing MACP2, the unbounded MACP2, you can see is fast moving. So this way we can differentiate the two populations. And by using super resolution microscope, we can actually trace the movement of each molecules and quantify their trajectory. And reconstruct that will give us two different categories. So the bounded MACP2 will give us a very tight spot here, meaning they are not moving at all. Well, the unbounded fractions will show these free moving patterns. So we can tell how much of the proteins are actually bounding to DNA in real time. So let me show you how it looks like in the real world. So we cultured mouse neurons carrying the wild type and mutant MACP2. And then under a microscope, here, this is showing the wild type molecules. And each of these very bright spot is one molecules of the protein. And you can see majority of them are actually staying at its original place. So they are not really moving because they are bound to the chromatin. Now, if we look at the mutant proteins, you can see they are freely moving. And you can also see that in the trajectory on the right. So we can actually quantify this difference, and we indeed saw a drastic and significant reductions of fractions of a bounded protein. And this is a really fantastic because right now we have a platform that allow us to quantitatively measure the DNA binding or chromatin dynamic of MACP2 in real time in live neurons. So you can imagine if we add a small molecule to these neurons and continuously measure the dynamic of these proteins, we can tell whether these molecules can increase the binding of MACP2, and that could potentially be developed into a drug. So after developing this platform, now we use human neurons obtained from the patient, and we are developing a high throughput screening system to systematically screen hundreds to thousands of small molecules, looking for candidates that can increase the binding of MACP2. Now, because we have the animal models, now we can further test the candidates in vivo and see whether that improves their behavior phenotype, which is the ground truth of the efficacy of the treatment. And eventually, hopefully, we can develop that into clinical trials, and we can test it in patients and potentially help them fight the disorders. So you can see in all cases, or in both cases, we started from the patient. And we use the animals to study the mechanism of the disease, while at the same time, we modeled the human cells to develop a platform that allow us to screen for drugs and small molecules, and we can further validate the efficacy of that drugs in the animal models. So all this would allow us to develop a better treatment that could potentially help the patient and improve their quality of life. So what I really learned by studying the behavior disorders is that by dissecting the interplay between transcription regulators, we can learn the mechanisms of how behaviors are regulated in both health and disease conditions. And I show you example of this new protein complex that genetically associated with a group of molecularly linked autism spectrum disorders. And by studying this group of molecularly associated disorders, we can find common disease mechanisms or pathway that would allow us to design targeted treatment to help the patient. Now, I think what's really important is that to going back and forth between humans and animal models. And we're always inspired by the patient, but we need to study them in the animals to look for the true phenotype. And it was the patient that allow us to find the mechanism of this complex and how they contribute to brain development. And it was the study of this disease that allow us to further develop a treatment that could potentially help those patients. So with that, I want to thank many people who actually contributed to this work. I want to thank my previous mentor, Huda Zogby, and other labs who helped us to accomplish these studies. And the work is supported by multiple foundings, including NIH, HHMI, Texas Children's Hospitals, and NDS. Okay. So I think before I end, I want to take this opportunity to really share with you that our new lab is now opening in Emory University at School of Medicine. So we will continue study the project that I showed you, and also we are developing new exciting project to study how neuroactivity contribute to brain disorders, and also try to find the link between sleep problems and autism and other neurodevelopmental disorders. And if you are really interested in this topic, welcome to join us, and let's do it together, right? So here I have shown you the QR code, and you can scan that and learn more about our labs. And thank you. With that, I'm happy to take any additional questions.